Welcome, everyone. Today, we are going to explore one of the most miraculous processes in all of biology, the phases of development. We will uncover how a single microscopic cell transforms into a fully formed, complex organism. It is a journey of precision, timing, and beautiful mathematics. Let us dive in. To begin our journey, imagine you are tasked with building a massive, complex skyscraper from absolute scratch. You can't just throw bricks into a pile and hope a building appears. You need a strict, step-by-step sequence of events. First, you need the architectural blueprints. Then you lay the foundation, followed by framing the walls, and finally installing the plumbing. If you try to install the roof before laying the foundation, the entire structure will collapse. Similarly, a living organism starts as a single microscopic cell that must transform into a complex being with trillions of cells. This miraculous transformation does not happen all at once, but rather unfolds through a highly coordinated series of steps. Now that we understand the need for a strict sequence, let us look at what these phases actually are. In developmental biology, the phases of development refer to the sequential, genetically programmed stages through which a single-celled zygote transforms into a fully formed, multicellular adult organism. Think of the zygote as the ultimate foundation. The genetic code acts as the blueprint, dictating exactly when and how cells should divide, move, and specialize. While development is a continuous, unbroken journey from conception to adulthood, biologists divide it into distinct major stages to better understand the specific milestones the embryo must achieve. Building on that definition, let us outline the specific stages of this journey. The process begins with fertilization, where the genetic material from two parents combines to form a unique, single-celled zygote. Next comes cleavage, which involves rapid cell divisions. Interestingly, during cleavage, the embryo does not grow in overall size. It simply partitions the large zygote into many smaller cells, called blastomeres. Following cleavage is gastrulation, a dramatic reorganization of these cells into 3 distinct germ layers. Finally, these layers interact during organogenesis to form the specific tissues and functioning organs of the body. Having seen the major stages, you might wonder why understanding this exact sequence is so critical. In the real world, this knowledge is the absolute foundation of reproductive medicine. It helps doctors treat infertility and ensure healthy pregnancies. Furthermore, when errors occur during these specific, highly regulated phases, they can lead to severe birth defects or developmental disorders. By studying the exact sequence of events, scientists can identify what goes wrong at a molecular level and develop interventions to prevent or treat these conditions. Without this regulation, chaotic cell division would result in a disorganized mass of tissue, not a living creature. Now that we appreciate the importance of these stages, let us zoom in on the cleavage phase specifically. Immediately after fertilization, the zygote must quickly build the cellular building blocks of the embryo. It does this through rapid, synchronous cell divisions. Because the embryo does not grow in total size during this time, the single large cell is simply subdivided into smaller and smaller cells. This process follows a geometric progression, meaning the number of cells doubles with every single division cycle. This explosive exponential growth is what allows the embryo to rapidly establish its foundation. This explosive growth we just discussed can be perfectly described using a simple mathematical formula. The formula is n sub t equals n sub 0 times 2 raised to the power of n. Let us break down what each symbol means. N sub t represents the total number of cells, or blastomeres, present in the embryo at a specific time. N sub 0 represents the initial number of cells, which is exactly 1, since we always start with a single-celled zygote. Finally, the variable n represents the number of cleavage divisions, or cell cycles, that have occurred. Because the base is 2, if the number of divisions increases, the total cell count increases exponentially. Now that we have our formula, let us see this with a concrete example. Suppose a frog zygote has just been fertilized and begins its rapid cleavage phase. We want to determine exactly how many cells will be present after it has completed 5 rounds of cell division. We know our initial cell count, n sub 0, is 1. The number of divisions, n, is 5. We substitute these values into our equation, giving us 1 times 2 to the 5th power. Calculating 2 to the 5th power gives us 32. This means the single-celled zygote has transformed into a 32-cell embryo, often called an early morula, because it resembles a small mulberry. Having calculated the number of cells, let us tackle a slightly more applied, real-world scenario involving the timing of these developmental phases. Imagine a marine biologist is studying sea urchin embryos in a laboratory incubator. The sea urchin zygote divides very rapidly, completing 1 full cleavage division every 30 minutes. The biologist needs to know how many hours it will take for the embryo to reach the blastula phase, which consists of 128 cells. To solve this, we must first find the number of divisions required and then calculate the total time. Let us work through the first part of the biologist's problem. We start by setting our target cell count, n sub t, to 128, and our initial cell count to 1. This gives us the equation 128 equals 1 times 2 to the power of n. Multiplying by 1 does not change the value, so we simply have 128 equals 2 to the power of n. By recognizing that 128 is a power of 2, we can rewrite it as 2 to the 7th power. Since the bases are exactly the same, we can directly equate the exponents. This tells us that exactly 7 divisions are required to reach the blastula stage. Now that we know it takes exactly 7 divisions, we must calculate the total time required for the entire process. We do this by multiplying the number of divisions by the duration of a single division cycle. The problem states that each division takes exactly 30 minutes to complete. Therefore, we simply multiply 7 by 30. This gives us a total time of 210 minutes. To make this number more intuitive, we divide by 60 to convert it into hours, resulting in 3.5 hours. The biologist must wait exactly 3.5 hours to observe the blastula stage, highlighting how mathematically predictable early development is. To bring all these concepts and calculations together, let us look at this visual representation of the developmental phases. On the far left, we start with the single-celled zygote formed during fertilization. As we move to the right, you can see the cleavage phase in action. The embryo divides into 2 cells, then 4, then 8, without growing in overall size. This rapid division leads to the hollow sphere of cells, known as the blastula. Finally, on the far right, we see gastrulation. The cells have dramatically reorganized into 3 distinct colored layers: the outer ectoderm, the middle mesoderm, and the inner endoderm. These layers will eventually form every organ in the body. To summarize our journey today, The phases of development represent a beautiful, highly orchestrated process. We start with fertilization to create the zygote and move through rapid cleavage divisions to build a multicellular foundation. Remember that this cleavage phase follows a strict geometric progression, allowing us to mathematically predict the number of cells at any given time. Following cleavage, the embryo undergoes gastrulation to establish the primary tissue layers, which then sculpt the final body through organogenesis. By mastering these sequential steps, you now possess the fundamental framework required to understand all of developmental biology. Great work today.